Doktor? Uh-huh.
Selamat pagi, good morning. Shall we
Ya
start?
doktor. Doktor Eric dan Doktor Vandy dia on the way.
On the way lah, okay.
Dia
Sebab
tengah
pukul
buka.
10.30 ada satu meeting lagi. Tak apa, saya tengah tunggu. So, we're waiting for Dr. Eric and Dr. Vanu coming in. I think maybe Sana, is Sana the first presenter now?
Ya doktor.
Hello, doctor.
Okay.
Hello. Yes, I'm the first.
Yeah, maybe Sana, you can try to upload your slide and then try to scroll up and down first, so we wouldn't get another additional time later. So you try to upload your slide. Are you okay?
Okay, doctor.
So we take another couple of minutes while waiting Dr. Eric and Dr. Vanu. Because 10:30 I have an appointment with the supplier about some instrument.
Hello, Dr. Hung, can you hear me?
Good morning, Dr. Venu. Yes, yes.
Yeah, good morning. Sorry, I have some issues of connecting. I've connected now
No
via
worries.
phone. Okay.
No worries, boss. And is Dr. Eric there?
Hold on.
Is Dr. Eric there?
No, I couldn't see him.
Dr. Eric belum masuk kan? Dr. Saida supposed to be chair for this section, but it's okay lah. No need to wait for another. We wait for Dr. Eric for another 1 minute.
Tadi Dr. Eric saya dah beritahu dia dah.
So tak apalah, kalau Dr. Venu sudah dekat sini, so kita boleh start dulu sebab student pun sudah tunggu lama dekat sini. Ataupun kena tunggu dia lagi.
Okay, Doktor.
Oh, kita mula sekejap, tak apalah sebab Dr. Eric dia pun tahu sikit. Nanti ada apa, yang paling penting slide tu dekat belakang lah. So I will chair the section lah. Wow.
Heng dia evaluator Dr. Eric itu.
Oh ya, Dr. Eric evaluator
Jadi kena tunggu dia.
Tunggu dia. Saya ingat dia chair ayo. Tak tahu. So Dr. Saida supposed to be chairperson, but dia tak ada kan? Dia tak ada.
Tukar petang, Doktor. So saya tolong dia untuk pagi ni.
Dia tolong pagi ni lah. Okay. So Bob nak chair ke apa?
Just tak chair pun just beritahu dia tunggu evaluator je.
Alright. So I will leave it to Bob lah. Thank you.
Ya, okay. Okay, Dr. Eric dah ada tu? Doktor?
Alright, kita boleh mula. Dr. Eric and Dr. Venu here, right?
Ya, sudah ada dua-dua.
Alright.
So, boleh, Sana can start now.
Kalau evaluator sudah sedia, kita boleh start lah.
Okay, I'm ready.
Okay, Sana can start now, your presentation.
Hello, Mr. Muhammad.
Hello?
Hello, doctor. Hello. Can I start?
Yeah, you can start now.
Okay. Hello, doctors. Hello, everyone. I'm Sena Yasin. This is my presentation. About my research progress, about my research project in this programs, master in mixed mode at Inform, characterization and immunoactivity of Phelinus lentus extract in macrophage cell line. Sorry. Table of content. This is my table of the content. Overview of the study and the focus of the area that I choose to do my research. Secondly, introduction about my research project. And thirdly, research question and also research methodology that I will go under the whole of my research project, and also data presentation and finding that I have working on it during this time till today, and the conclusion and Gantt chart timeline of my research of my master's degree and research project. What's the focus and the interesting area of this research project that I took with under the supervision of Dr. Leo? The one it's all about medicinal mushroom. Already medicinal mushroom or mushroom have long been suggested for impacting immunity, cancer, and autoimmunity, and also like being used as a traditional medicine by China and middle like in East Asia and also in Europe countries too. But recently, scientific research is now focusing on identifying specific bioactive compounds and their mechanisms of action through their extraction. So they are looking for and focusing on the specific bioactive compounds from the crude extract that we are extracting from the mushrooms. And this is the Phelinus lintus, the species that I will study in my research project. Introduction and background about my study. Firstly, the background of my studies came from the raising demand for immuno-enhancing products and the product that enhancing or boosting immune system, and also like in the literature. They have been known since 2022, known as immunoceutical under the nutraceutical products. Driven by aging population and the prevalence of infectious disease, there is a significant global interest in foods and medicines that boost immune functions. This is also concluded with the WHO reports that have been studied and published in 2022. Like they studied that thing that the aging population will be doubled by 2015. The importance of my study, I will study the role of medicinal mushroom as immunoactivity or as immunoceutical product. So medicinal mushroom, especially the one like reishi, shiitake, and maitake, have been recognized for their immunoboosting property for centuries, and they have gone under the clinical trials, phase 1, 2, and 3, and they have been using in the community. Their bioactive compounds like beta-glucans and tryptanoids play a crucial role in enhancing immune response and reducing inflammation. So they have been using as a immunoproduct to boosting the immune system, enhancing or suppressing immune system. About the literature review, that focus on the chosen species of my study, Phelinus lintus. It's firstly Phelinus lintus. It's a rare mushroom that grows on Mars elbow, mulberry tree in China, Japan, Korea, and also in America and Africa. Exhibits many bioactivities, especially the one that goes under anti-cancer, immunomodulatory, and anti-inflammatory effects. And these effects came from their bioactive compounds that have been studied in the literature and published. That the one contains polysaccharides, phenol, propanoids, flavonoids, phenolic compounds, furan, and terpenoid. So all the activities, especially the immunoactivity, goes under to these bioactive compounds that have been found in the polysaccharide or the crude extract of this Phelinus lintus, the one that found in the nature. So what's the connection of Phelinus lintus, how it enhances immune system? So during all the literature review of all the published papers that have been studied, mostly they worked on the macrophage cell line, but the macrophage cell line from the murine example, murine models, the one raw macrophage cells that we found like its effect, it goes under the macrophage activation polarization, phagocytic activity, and T cell activations. This is Phelinus lintus. fruit like found naturally founding on the mulberry tree in China or Japan or Korea, and this is their fresh appearance, like yellow and like submerged in the tree. And this is the polysaccharide structure and also furan structures that is one of the common, two of the common compounds in the crude extract of this Phyllines lentus. Phyllines lentus, its taxonomy, it goes under the fungi, kingdom of the fungi, and Basidomyces, Basidomycota phylum, and also its genus Phyllines, QL, and its species, the one that we will study, it's Phyllines lentus. This is its appearance and its morphology. Firstly, like it's a perennial fruiting body. It has perennial or flat fruiting body, and its mycelium submerged or merged in the tree, and it cannot be differentiated. It cannot be see it like clearly as other types of the mushrooms. This part it's a fruiting body, and this part it's mycelium. And its color is brown surface when fresh and turning to black when it became drying and old. But it's inside it's cinnamon yellowish brown dry tubes, and mostly known as a horseshoe like appearance, or has that appearance. About my research question, my research question of this research project, it's how does the crude polysaccharide from the ferment, everything that have been done in the literature, it's all about naturally founded, natural growing Phyllines lentus. But what about my study? My study is about how does the crude polysaccharide from fermented Phyllines lentus using hot water extraction influence antioxidant activity, or like generally modulate macrophage cell line. And the macrophage cell line that have been used, we will use in this study, it's gonna be human-based monocyte THP1 cells. And hypothesis of this study, bioactive compounds in crude extract from whole solid fermented Phyllines lentus will significantly modulate human macrophage cell line THP1 immunoresponses, as how it happened with the naturally founded, naturally growing Phyllines lentus. My research methodology, first part of my research methodology, or the methodology of this research, is going to be crude polysaccharide extraction preparation. Firstly, to prepare the crude polysaccharide extraction, first, the most important one, identification of Phyllines lentus strains through the sequencing, DNA extraction and sequencing, to be sure about the species and strains of this fungi that we are studying, the one that have been going under the solid fermentation. And secondly, hot water extraction, extracting the compounds from that solid fermented Phyllines lentus that we have collected in the we have collected in the industry. Then characterization of the crude extract through the colorimetric assay, and polysaccharide, beta-glucan, tretinoid, totalflavonoid, and totalflavonoid. These are the compounds that we will do the characterization for them to find out about their percentage in the crude extract. And the second part of this methodology is going to go under the in vitro immunoassay through the help of the THP1 cell monocyte cell line, human monocyte cell line. Firstly, antioxidant activity of the crude extract, and through the DPPH assay, ABTS, and FRAP assay. And then go to the cell assay in vitro cell viability test, MTS assay, to define the concentration level with the minimum cytotoxicity level. Immunoactivity measurement, it goes with it's going to be done with the real-time PCR through the cytokine gene expression profile, studying this gene expression profiles of the cytokine that have been secreted through the activation of the macrophage cell line. Immunocell activity, we will do the nitric oxide or NO production after the cell being treated with the crude polysaccharides. Data presentation and finding of my research till today. First of all, sample collection. The first one, we have Phalaenus linters that grow on organic substrate using solid fermentation technology. And we are using this sample for ID identification or DNA sequencing to know it's exactly the Phalaenus linters or other species of the Phalaenus linters. Dried and pulverized sample of the fermented Phalaenus linters sample after having grown on the solid substrate or organic substrates like beans, rice, and then go under the solid fermentation and dried out and pulverized sample that we have in lab, and we will work on this sample through hot water extraction and producing crude polysaccharide extract. ID identification will go under the several steps. Firstly, DNA extraction from fungi, the fresh grown one. Secondly, PCR amplification of the product of the DNA extract, and then DNA gel electrophoresis, purification of DNA or PCR products, and then sending this purified DNA to the DNA sequencing and analyzing the data of DNA sequencing through the bioinformatic analysis. DNA extraction and amplification. Mushroom DNA identifications. We selected one. The first one, the A, it's a Phalaenus linters, the one that we collected from the fresh growth cultures. And from B to F, it goes to the positive control, other medicinal mushrooms that have been studied before. DNA extraction, we will use the kit. It's called Easy Pure DNA Extraction Kit from Transgene biotech company from China. And primer, the primer that have been chosen, it's universally primer for fungi or medicinal fungi. Medicinal mushroom ITS primer, Pierce primers. It's called internal transcribable spacer. This is the location of the primer for the amplification and sequencing of ITS region. It's forward and it's reverse. PCR amplification through the Agilent Sur Cycler 88000 Agilent US device, and it go under the 35 cycle. First phase is denaturation for 30 seconds at 94 degrees Celsius and 55 degrees Celsius for annealing. There is missing annealing, and then for 1 minute, and then extension at 72 degrees Celsius for 40 seconds. This is the result that I got through my working in 1 week. Gel electrophoresis result that I got. Unfortunately, from the first try that I didn't get the band for my sample Phalaenus linters, the A1, and only three of my positive control I got the bands, and the one that with no bands. The N1 it's for positive for negative control, and from B to F it's positive control. Only three of them, B, D, and E. I got the result, and this is my result. Sorry. Ongoing lab work right now that I'm working on the fermented Phalaenus linters dried powder extraction that goes under the hot water extraction through the bath, through the water bath, and crude extract alcohol precipitation by ethanol 95%, and then dry freezing my product from the extraction and precipitation, and then characterizing this precipitated and freeze-dried compounds to be studied for to go under the study of its characterization. Conclusion of my study. Firstly, I know, my result, it's not enough to be represented, but this is all the result that I got from the 2 weeks of working on DNA identification and DNA extraction. So my conclusion is about first, like the steps that I took, it's DNA extraction, amplification, and gel electrophoresis. And the result only I got positive controls, showed 3 bands, the bands of the positive control, and the sample strain didn't produce the expected results. The current effort that I'm working on it, troubleshooting my experiment, optimizing the method that I use it and follow it, and also finding out new methods and optimizing the method that I was working on it, repeating experiments to get the bands, or optimizing the methods and then repeating the experiment this week and next week, inshallah. And the extraction work, I just will start in this week, producing fruit polysaccharide from mushroom for in vitro assay. This is the work for the showcase in lab that I just worked in these 2 weeks. And this is my Gantt chart. As you can see, like my proposal writing, it took 2 months, and the submission, it's the end of the June, and also like my work just started in the mid of June. So this is all my progress that I have till today. This is my references, and thank you so much for your time and your listening.
Hi, Sana.
Yes.
Thank you so much. Very nice presentation. Okay. Can you go to your research question?
Research question? Sorry,
Yes.
sorry. Yes.
Yes. Okay. The research, what I want to ask you is that you have been keep on talking on your literature review and everything about the compound and so on, but
Sorry,
you have planned to use the fermented part of it. So is there anyone have used any other compound which is fermented and they got some results?
Sorry, doctor, like I cannot hear you. The problem of my laptop just... Can you just come up with that question again?
Yeah, can you hear me now?
I cannot hear you that much clearly.
Okay, hold on.
Okay, doctor.
Hi, Sana, can you hear me now?
Hello, doctor.
Yeah, can you hear me clearly?
Yes.
Okay. The question that I asked is, in your introduction and literature review, you were talking about the Pilanes linters, extracts, and so on. But the work that you're going to do is you are going to take the fermented, okay, part
Yes.
of it. So is there any other literature review or for different plants, if it is not for Pilanes linters, that they have used the fermented part of the product and they have get some data on the inflammatory?
Doctor, like in the first, like I didn't go under the studying and go through the literature of the fermented one, but the way that when I just did my thing and knew about what I'm doing, so the literature says, but I cannot say like with this only one just generally like a review on, saying like with the fermented one on the organic substrate, it helped to enhance increasing the secondary metabolite or metabolite of the fungi mushrooms through their producing the more enzymes and the secondary metabolite of this organisms like mushrooms, because there is organic compounds helping with the nutrition to the mushrooms to produce this kind of the enzymes.
Okay, so I think this part of it, you should highlight it when next time you're presenting that the
Yes.
fermented part of it. So what does this, what
Yes.
it can do and what it does and where's the literature review of it, so that when you come and say that you want to use the fermented part of it, so we have an idea that why is the reason that you want to use the fermented part. Okay?
Yes. Understood, Doctor.
Okay. So can you go to your objective?
Yes. I didn't provide my objective, Doctor.
Yeah, so that's why I'm asking, because you need to provide the objective so that we would know, because when you're presenting, so what is your objective and how far you are now and where that you need to go. So without your objective, it is not clear that, so now you're saying that you have identified and you're going to do the next part, you're doing DNA extraction and so on. So I'm not sure that where you'd stop without the research objective. So where that you would go.
True. Doctor, like they provided a guideline for us. I just when I went through, there wasn't any objective, so I just like escaped that one. This is one of the reasons that I didn't put my objective.
Okay. Next time, I think better you put. And then I think you also didn't show your research flow, right?
Yes, Doctor.
Yeah. So we need to also.
Yeah,
so can I see the flow? Yeah. No. Yeah. So this is your flow of the work.
Yes, Doctor.
Okay. So you are now the yellow color, the blue you have completed, the red you haven't completed. Am I correct?
Yes, Doctor. Because if I cannot, because the first one, the blue one, I have to finish and I have to have my products, my crude, characterized polysaccharide, the crude polysaccharide, and then I can go to my second part of my research.
Okay. So you said that you've just started your work in June, early
Yes,
June.
Doctor.
So you now you have completed the characterization?
No, Doctor. Like I'm just like I started like mid of June, like after our proposal presentation. So right now that's why I don't have enough results. Maybe only the gel electrophoresis, and that's why like I'm just in the beginning of my research.
Okay. I think you have to, because looking at your flow chart, you need to speed up a bit so that you can complete your antioxidant, because you have antioxidant, cell assay, and also your RT-PCR. Okay.
Yes, sir. If you can see like from my Gantt chart, it's just like from the end of the June, ID identification of DNA sequencing and extraction, and starting with my characterization, it's gonna take like 3 months, mostly to finish all these steps of my research.
Yeah, I understand that, because you have put here your cell viability MTT 1 month, okay, and so 1 month, but sometimes it'll take more than that. So so try to speed up a bit. It's just an advice outside of here, so that when you speed up a bit, then in case you need to repeat some work or something, you'll have enough time. Okay.
Okay, Doctor. Thank you so much.
Okay. Yeah. So that's all from me.
Thank you so much.
Thank you, Sana.
Yes, Doctor.
Can you go back to the gel electrophoresis?
Yes, Doctor.
So, which one is DNA? Like DNA is which one?
DNA, Doctor.
Yeah, you want to extract to identify which is which batch.
Doctor, like this is as from this workflow, I had 6 samples. One of the sample is my species, and the other one is all positive control of fungi that I've been using for comparing and for my study. So, but from my result, only I got 3 of my positive control, the bands of them. And unfortunately, I didn't get the band for my sample. So in that case, like I have to repeat it again. And also like before repeating, I have to do the troubleshooting and optimizing the method that I use it.
Okay, but bit strange. What's why you have so many positive control?
Sorry, doctor.
Why you have so many positive control? Do you have many different type of strain or only one? This is come from the B until F, the positive control, right? So this is come from different strain or what? Different species or what? Or it's just
No,
come from
it's doctor, it's a different species, but they have the same region. They just like use it in case for the positive control to have. Right now, I have 3 positive control. So in that case, my aim is gonna be only like getting my species strain back.
maybe I'm not sure. Yeah. Because you have to remember your objective is really to get the A out right, identify the A through the sequencings, not so the B and the F. Why so many positive control? You need so many positive control. That's why my question lah. I go back and maybe discuss with supervisor. Usually when you do this kind of things, you must have maybe duplicate, triplicate, or this kind of your things. One time only, you just do one time with one condition, quite difficult to get. And you so many positive control. Not sure. And the positive control, the CF also don't have, right? Why? Why
Yes,
the
doctor.
CF
Yeah, I
also don't
didn't
have?
get that.
Shana, I'm not sure. Is one thing I want to ask, is this necessary to do the ID identification for this project?
Yes, doctor, because like when the Felinus linters go under the solid fermentation,
Okay.
the ID already like in the literature, the ID of Felinus linters is just there is a question mark about its like real identity because it has like similarity with other species. So to make sure like this species that you are studying, it's Felinus linters, so you have to do that identification, especially the one like use it for the fermentation.
Okay. Just in case you didn't get
Yes.
any identification for extraction, you fail to get the ID, what you gonna do? Because this one you cannot finish the ID, then you cannot proceed for the next. What you gonna do with this? Are you gonna try more hard? I'm not sure. This is first time you only try one condition or what? Because this is PCR, right? You only
Yes,
try
doctor.
one condition only?
Yeah, it's this is PCR, doctor. Like I tried, I did like repeated my gelatin for this 3 sample, my sample and 2 other positive control, but I didn't get the band. So these 2 times I just like because I am the first, like this is my first time to work in the lab, so just troubleshooting with my like skills in the pipetting and how I handle it and pipetted the sample, but it didn't work. So it means like I have to go to the optimizing the method that I follow it.
Okay. I'm not
Yes.
sure lah, actually. Since you insist for me, not really. But since you insist for the ID identifications, then okay lah. Since you agree, never mind. I think you try harder because it's not necessarily the same condition will work out for everything. This one, I think you try with same condition PCR parameter with all the ABC and F, right? Or you use the same condition or different condition? You mean the temperature?
The same condition, doctor, because like for all the samples we are using the same primer.
Oh, okay. So I think you have to check different parameter and different since working your BDE is working on positive, it means your pipetting is usually not an issue lah. what you usually do. If all is not working, maybe you pipetting the issue. Since 3 of the is working, pipetting should be not the issue. Maybe I think you have to check your parameter actually. Not sure
Yes,
this
doctor.
time.
Yes.
Because the same condition you apply for everything. I mean, shouldn't the other thing should work Maybe I think sometimes the primer also the big concern also usually because you use different primer, right? You use
Yes.
different primer or same primer? Different primer.
Sorry, doctor.
You use same primer or different primer? Different set of
The
primer.
same primer, doctor, because this one is a universal
okay.
primer for fungi.
Okay, use universal primer this one.
Yes.
Okay. I'm not sure because if I see the project, I will more emphasize on the later stage when you have all the tests. This one, I'm not sure why you want to do the ID applications, but let's all the test is far more important because this one you can you cannot proceed this project without the identification or what?
This is one of the necessary, doctor. The one that I said, because the sample that I have, the powder, it's the same that I took from the growth, the fruiting body and mycelium of the one that I did DNA extraction. So to make sure, okay, it says like this one Philines lentus, but because when you do the fermentation and use it for the fermentation, there is already question about this, this, there's a gap about, is it the same Philines lentus?
Why you cannot do the test finish it before you do that? You get the ID because you stuck here forever. You cannot get the ID. Then how? Let's say you get the ID. What you gonna do? Say because you use the same primer, right? You use the same primer for all the things. So what you gonna prove? This why I question. So why you gonna prove? Because same primer, what you say what? A get use the same primer, B a same primer. A is your sample, B until F is same primer. So why I don't get it actually? Why what so? This is ID, right? After that, you get the thing, or you want to send for sequencing. I'm not sure what.
Yes, it will go to the under sequencing, and then after sequencing with the bioinformatic analysis, do the phylogenetic tree to find out about the species and the genus and the species of the from that sample.
Okay, since you insist on it, I'm not sure because there are so many essay at the back there that you want to do. Like Dr. Medu say, we might take some time also to finish the thing. So you stuck on you've been settled in the. I'm not sure. This is maybe first time you do. Maybe you can get in the second try already. Think other than that, I don't have other issue. I think you need to put objective okay. You
Okay.
need to put objective. I think the first time we see people without objective inside, stated objective. Okay, you need to put the people questioning with objective. Okay, that's my questions. I think thank you very much.
Thank you so much, doctor. Thank you for your.
Chairman, again, I don't have question anymore. Anyone?
Okay, we can go to the next student. Peter, you here, Peter?
Yes, I'm here.
Evaluator Dr. Eugene tak masuk lagi. I already message him. Right. So, tunggu kejap. Dr. Eugene in already. Mr. Chairperson, please start now. Thank you. Sorry for the delay.
Okay, Dr. Eugene start. Maybe we can start now. Peter?
Okay, everyone can see my slides. Yes, Jinta, you can proceed. Okay. So my presentation topic is pentapeptide identification for the inhibition between category B protein by in silico approaches. My name is Jintao Pan and my supervisor is Dr. Chan. Okay, today they will have the 7 part of my presentation. Next. So S189 monomer looks like M shape when it is in homodimeric situation, including the 4 helix and 3 hitch regions. And S189 is assembling to amylose, not only self-assembling but also assembling with other members of S189, such as S100AA or S112. And S189 amylose cause lots of disease, regional toxicity, and AD or cancers. So S189 self-assembling is relevant with many diseases. And to block S189 self-assembly is required to identify novel peptide drugs that are more biologically friendly and have stronger binding ability. Building a virtual peptide library makes it simple to uncover prospective medications for treating not only S189 self-assembling, but also additional disorders in the future. So there are 3 purposes. First one, to construct a 5-membered peptide library for virtual sequencing. 2, to identify the peptides that can bind with S189. And last one is to evaluate the interaction between pentapeptide and S189. Okay, the cost of the peptide production generation increase with the length of the amino acid chains, such as peptide synthesis company calculate the price of the peptide producing based on number of amino acids and concentration. So more regions may signify higher binding capacity, but longer peptides are also more likely to form second structure. Although longer peptides give us with more options, and the number of peptides increase 20-fold when each amino acid is added. So this is the reason why we choose to build the pentapeptide library. It's one of the literature review. There are the 3 S189 inhibitors can block the animal aggregation of S189. ApoE is 299 amino acids longer than S189 homodimer, which is 228 amino acids. Therefore, it appears to be a big clip used to clap the S189 homodimer in solution and restrict its attention. And research of polysimultative lattice designed the sections of S189 from LYS 50 to LYS 54 as have a high annual propensity. And they describe the S189 amylo connection line occurs between both ends of each S189 just like this one. And it was mentioned that only a naturally occurring chemical in olive oil has the ability to disaggregate S189 fibers on the base of blocking the elongation of exciting S189 fibers. So ODA shows peptide stack with TRP88 hydrogen bonds with ARG85 and GLU552 of S189. The interaction with S189 will includes the high amylopropensive section from LYS50 to LYS54. Serpent liability to disaggregate may be the result of an interaction force with the area from ARG55 to TRP88. So we think this area from ARG55 to LA89 and from LEU49 to VL55 of 189 is active as area for S189 amylose formation. So it's my methods and data collection. Some 3D structure of molecules will be done from the protein data bank and ChemiSpider database. ODA would be my positive control to compare the binding ability of peptides. And producing the pentapeptide library via RDKIT Python API. It can transfer a peptide sequence to PDB format files. An open Babel will be used in converting the PDB to PDBQT, because the PDBQT is the special format used in Autotalk. And binding free energy and hydrogen bond will be analyzed and then ranking the Autotalk results. The most detailed analysis would be use the AMBER 2080 to do molecular dynamic simulation. So it's my results. First of thing, in this research, we use arrangement and combine of 20 amino acids in two sites results to 400 results, and then putting code into the 400 results. By this way, we can produce any peptide library when change the code putting in this method. For example, we can put the same 400 results into the middle of the 400 results and generate the tetrapeptide library, including 160,000 peptides. And then the pentapeptide library can be achieved by putting the 20 amino acids into middle of each peptide in tetrapeptide library. So the 20 multiply 160,000 equal to 3,200,000. And then the safe paths such as C and ACD and then AACDA PDB. So my view is to minimize the sample we do at the same time. So I separate the whole pentapeptide library into some small group. So usually I only use the 400 peptides in each time. Next. I use the RDKit API. I spent 10 days to build the pentapeptide library. About the RDKit, there are two sub-packages, more from sequence and more to PDB block. So the first one, they can constructing a molecule from sequencing strings. The next one, they will save the molecule to PDB file format. And then we spend 30 hours on converting PDB to PDB-QT form via OpenBeba API. However, when use CMD code command in Windows to use OpenBeba, the same number of files would take 22 hours. And then the total size of the pentapeptide library in PDB format is the 28.7 GB. And the pentapeptide library already was released online. After build the pentapeptide library, the next thing is to reproduce the docking results of S100A9 and ODA, because the ODA is my positive control. And after reproduce the docking results, and I can identify the docking center and other parameters to do to screen my pentapeptide library. So because the RTK didn't update the docking results of ODA with 100A9, so we choose the similar area with results of attacker to identify identify the docking center, and then use docking autodock to docking 300 times between ODA with S100A9. And these are the docking results of ODA with S100A9. We reproduce the docking results between ODA with S100A9 to be my positive control. The docking results with top 10 lower binding free energy. It suggests that ODA tends to create a peptide stick with trypsin 88 and hydrogen bonds with ARG 85 and U52 of S100A9. Our results is similar with the results of the article. Hence, the geometric center coordinates of the ODA structure with the lower binding free energy. This figure A is used as docking center in further docking. And then I randomly choose 25 member peptides to find a gray box size. You can see the 50 size is not enough to wrap all of peptides, so I choose the 60 size. But it means we should spend 4 minutes in each pentapeptide via docking. So there is a big problem. We don't have enough resources to docking our pentapeptide library. So for saving time of screening, we docking the tripeptide library with S100A9 first, and then choose the triple peptide with high score of binding free energy and score to isolate the pentapeptide library. We can put the triple peptide in the middle and the left and the right. And then we will have so we use the lost pentapeptide. The tripeptide library only 8,000 peptides, and each tripeptide corresponds to 1,200 pentapeptides. This is the result of docking between triple peptide with S100A9. Is this distribution of binding free energy. You can see the three tripeptides, WWF, WPW, and YWF, is binding free energy near negative 8.0 kcal per mole. which is close to ODA's binding free energy of negative 8.23 kcal per mole. And then we can use the XMO to see the structure. And all three peptide with the first three high binding free energy with S100Ni can form peptide stick with trypsin 88 of S100Ni and hydrogen bond with ARG85 and other regions such as ALA89 or HIS 105. And especially with the force posture of WWF peptide, is based peptide stick with trypsin 88 and hydrogen bond with ARG85 and 0U55 of S100A9, similar to ODA. So we choose the three peptides as code to isolate the pentapeptide for further selection. Okay, next. So it spent 10 days on docking 3,099 599 pentapeptides with S100A9 via autodock villa. Comparing with the triple peptide, the pentapeptides tends to form lower binding free energy, that means stroke binding. And pentapeptide with WWF and WPW code may be bind to S100A9 more effectively than with YWF. Because pentapeptide with WWF and WPW codes are more likely to provide a large number of outcomes with a lower binding free energy. And especially those pentapeptide including WPWF, you can see there are two docking results lower than negative 9 kcal per mole. There are many pentapeptide has lower binding free energy with S100A9 than ODA, and there are 7 pentapeptide with binding free energy less or equal to negative 80.7 kcal per mole. And WPW TY with the best binding free energy by binding outside of active site. So we choose the second one, the WWPWH with into the MD simulation process. And the second posture of the WPWYW peptide with binding free energy negative 8.7 has the greatest number of hydrogen bonds with S100A9. There are 7 hydrogen bonds. And actually, when the binding free energy difference is only 0.1 kcal per mole, they are little different. So the WPWYW peptide was chosen for further MD simulation. Okay, so we successfully identified 2 pentapeptide with potential to inhibit the S100A9 assembly. They not only with lower binding free energy than ODA, but also have more interaction with S100A9. And then we go through MD simulation to discover more detail of the interaction with S100A9. And about the conclusion, we already built the pentapeptide library, and it was released on USM library, as well identify 2 potential treatment pentapeptides. So the top 2 purpose we already finished, and last one is to elucidate the interaction between pentapeptide and S100A9. About the guide I already finished 2 steps, the peptide library and peptide selection by docking tools. So I will have 4 months to do the MD simulation. I will learn how to use the Amber 80 during this break, and then start MD simulation on October. Okay, that's my reference. Thank you for your time. Hello?
Okay, thank you, Jintao. Since Dr. Ong didn't mention, maybe he stepped away, so I will just start first. So you started screening with the peptide library, but you mentioned that it is maybe taking too much time and resources, so you started with the 3-peptide library.
Yeah.
For the 3-peptide library, is it also the 3.2 million one that or is it different number? Because maybe I might have missed it.
Oh, it's only the three peptide library only 8,000.
10,000?
8.
8,000. Oh, okay. Yeah. Okay. So from 8,000, you were able to identify how many candidates from here?
3 is near the 8.0 kcal per mole.
Oh, okay.
I choose the top 3.
You
Actually,
chose
there are
the
so
top
many,
3.
so I choose the top 3. I want more choices. Yeah.
Okay. But did you investigate the others besides the top 3?
Yeah, actually, I think most of them can have some hydrogen bonds with the similar active areas with the ODA.
So would you say because there is a lot of W recurring or repeats, we can see WP, WWF. So is the tryptophan residue important in this case?
I'm not sure, so I need to do MD simulation to discussion that.
Okay.
Because how to say that? For this result, you can see they can form. Yeah, you can see they have pivot stick with the ATA. So they need the I don't know how to say that in English. they need to need this structure to have
Okay.
to compare the pivot stick with this
Okay.
ATA. So
Okay.
this is the reason why the W is very important.
Yeah. So they need the sort of bulky structure, right?
Yeah.
Okay. And then in slide 10, you showed the docking. In slide 10, slide number 10. Yes. Okay. So at first you try to reproduce this. And I'm just wondering, because you said this is the control inhibitor.
Yeah.
Yeah, but from what I see is that the OLEA is docked to the out